Alright class, so here's our last lecture video on inheritance in genetics. So far we've done simple, basic, complete dominance, and then incomplete dominance where we see a mix of phenotypes. So now this last set of problems, it's actually going to follow a couple of patterns here. So one's called codominance, and this actually means that we're going to have two dominant alleles. Right, so not just one dominant allele, but two dominant alleles. And we are also still going to have one recessive allele. But the name codominance, like co is together, right? Like cooperative, together. So two dominant alleles still have a recessive, but the important difference is two dominant alleles. And these two dominant alleles are equally expressed. And I'm getting rid of my one recessive allele comment here because I don't want you to think that the recessive allele is included in that. Recessive is always recessive. So the two dominant alleles are equally expressed. That's codominance. And then multiple alleles is just telling us that we have more than two possible alleles. More than two possible alleles. So in this case we have three because we've got two dominants and a recessive. So we would have three kinds of alleles. Alright, so in all of these, the character that we're going to be working with is the ABO blood types in humans. The ABO blood types in humans. So then the traits are the actual four blood types, right? Type A, type B, type AB, and type O. And then I have to mention here, because I know lots of people are going to see this and watch this video, and right now you're thinking, well, what about positive, negative? Because we hear those with our blood types, right? B positive, O negative, whatever. Well, that's a separate character. So if we included that, we would be following the inheritance of two characters, not just one. And that gets a little more complicated than what we're looking at here. Okay, so positive, negative, separate character, separate thing. Here we're just looking at A's and B's and O, I guess, if we don't have A's and B's, just O. Right, so those are our traits. And then I want to show you the alleles that we have. You know, this can be, this is a little more specific than what we've seen. Like so far we're using things like, just use A's or B's or whatever. These are really specific, and it just needs to be to make this work out correctly. So three alleles, two are dominant, so big I, and then superscript A or superscript B, so it's IA or IB, and then there is a recessive allele, little I. Okay, and then I want to make a table here for you. So if we look at a specific genotype, then what blood types do we get? Okay, so some of these are, pretty straightforward, make sense. Like if we have IA and IA, that's got to be type A, right? Because they both say A, so there's no other option. But we can also get type A blood if we have IA and little I. Because that's just regular O complete dominance, right? There's a dominant allele and a recessive allele. How do we know? One's uppercase, one's lowercase. So either of those genotypes would give us type A blood. And then we can see the same thing but with B. IB, IB, and IB with a little I. Either of those gives you type B blood. So we will never, in the problems we're about to work and problems you would see from me with these, like I can't simply just say cross a parent that has type A blood with whatever. Because if it just said type A blood, you would come back to this table and you would say, well, which one of these would I use? And it's not random. I mean, that person doesn't have both of those things, and that person can't have both of those things. It's got to be one or the other. So I will tell you specifically, cross this with that if it's A or B. Hey, and remember our terminology we did in our very first lecture. If these alleles are the same, like this one here or this one here, the alleles are the same, homozygous. If the alleles are different, heterozygous. So we would still use that terminology. It would be this person is homozygous for type B blood. That would tell you exactly which one to use. All right, we can have our two dominant alleles together, IA, IB. So this is actually the only thing here that's codominance. Right? Two dominant alleles together, equally expressed, type AB blood. It's basically like having type A blood and type B blood both at the same time. And then our last option is if we're just homozygous recessive, right? What if we have both little i's? And that is type O. Right? So two little i's is basically telling you there's no A's, there's no B's. That's type O. Right? So type O sort of actually isn't like because of something that's there, it's because of stuff that's not there. There's no A's, there's no B's. All right, so you'll want to refer back to this table, working problems on blood type crosses that we're doing. And I encourage you, look back at the table, use the table, because I'm going to erase this, so you don't need to add this to your notes. But the problem is, like, if we end up saying, hey, let's cross homozygous B with type O. You know, if you're not using genotypes like this, then you're going to do something like, okay, homozygous B with type O. And then you do some kind of Punnett square that's going to, I mean, all right, so what is that? BO, exactly. That's just like, that's nothing. I don't know what that means. There's no way of knowing what that means because it literally means nothing. So don't just guess and use whatever. Use these things in this, these alleles, these genotypes that I'm giving you. All right, so first practice, let's cross heterozygous A with heterozygous B. So we will rewrite heterozygous A, heterozygous B. Nothing's changed on how to do this, right? So gametes, figure those out the same way. Sorry, I used the comma there instead of the or. It doesn't matter. 2 by 2 Punnett square. You want to know if you're doing these correctly, pause this lecture video, work them on your own, come back to it in a minute or so, see if you did it right. Filling in the Punnett square. been sitting here doing videos for a while, so my handwriting's getting really tired and sloppy. All right, so we've filled in our Punnett square. So now ratio for this, we'll do this a little different. So with this, what we'll say is something like, what are the chances? Remember, these are offspring, right? So since we're doing human ABO blood types, like these are possible genotypes that this couple's baby could have. So what we're looking at then, what we want to know are the chances that the baby has, and then the 4 blood types, right? Type O, or sorry, type A, type B, type AB, type O. And I would do these as percentages. I know we've done everything as fractions so far, but I would just do them as percentages. So we come back to the Punnett square, we count. How many would be A? Okay, well, this would be AB, this would be A, this would be B, this would be O. So how many are A? 1 out of 4, 25%. How many are B? 1 out of 4, 25%. AB, 1 out of 4, 25%. O, 1 out of 4, 25%. All right, so that's what we're going to do with ratio stuff for the blood type. So we're not technically doing like a genotypic ratio. This is a phenotypic ratio. It's just a different way of asking you to do it. All right, so that's our ratios that we're doing. And I started with this, you know, you often in classes get questions about, well, if I have this type of blood and my husband has that type of blood, how can our kid have, you get a lot of those questions. And I mean, look at what we crossed here, right? If we didn't know all these genetics, we're crossing A with B, right? And a lot of people think then, like, if I'm A and he's B and we have a kid that has type O, like how's that work? I mean, one's A and one's B and the kid is O. Like, that doesn't seem like it works. But here's A crossed with B, both heterozygous, and we can get 25% with type O blood. So it depends on the actual genetics, right? It depends on the genotype that we're starting with. All right, so now let's cross homozygous for A with type AB. Another good chance to pause, complete this on your own, come back to it, check your work. All right, so we're doing homozygous A cross with AB. We want to know our gametes. We're doing 2 by 2 Punnett square. All right, fill it in. And then we're doing our percent chances for each blood type. All right, so this one would be A, this one would be AB, this one is A, this one is AB. But still, we're going to do this whole list. So 2 of them are A, so 2 out of 4, 50%. None of them are B, right? 0%. And the thing with this is, like when you see quizzes for this stuff, like it may be a multiple choice question that says, what percent chance can this child have of having type A or type B blood? And if you only do something like this, A is 50%, AB is 50%, and you see a question that says, what's the chance they can have type B blood? I mean, maybe you come back to this and you're like, oh, well, there's no B in there, so it's 0. But there's going to be a lot of people that come back to this and go, I don't understand. You're asking about B, but there's no B on my list. So, I mean, you can work them the way you want. I'm not sitting there supervising every cross you're doing, but I think it's easier to have all of this listed. So AB is 2 out of 4, 50%. O is 0 out of 4, 50. 0 out of 4 is 50%. 0 out of 4, 0%. All right, so that's ABO blood types. Codominance, multiple alleles.